Welcome back, students. In this section and from this section onwards, I am going to talk to you about protein metabolism in terms of the different carbon skeletons of different amino acids. So I have divided this into, when you look at an individual amino acid, it has got its metabolism that is there, whereby it may be glucogenic or ketogenic, or it may be the synthesis of the non-essential amino acids. Remember, we cannot synthesize the essential amino acids. But how are the non-essential amino acids synthesized within the body? I'll be briefly touching upon it. And we look at the biological importance of these amino acids. How do they help in the formation of what product? The biological importance. And certain disorders which are associated with the amino acid metabolism. So I'll be dealing with each of these separately. So let us look at the catabolism of an amino acid. One thing you should be knowing by now. It has an amino group and a carbon skeleton. So removal of the alpha amino group is as urea. Yes, you have told urea, then you are right. So it is disposal of nitrogen in the form of urea. Removal of the alpha amino group. We come to the metabolism of the carbon skeleton. This can be converged. We can always say, I told you about glucogenic and ketogenic. Now I go further. Now all this metabolism can result in the formation of 7 products. Okay, they all converge to form 7 products. Which are they? This makes your job easier. Only 7 are there. These are oxaloacetate. 2 amino acids form oxaloacetates. 5 amino acids form alpha-ketoglutarate, 6 form pyruvate, 2 form fumarate, and succinyl-CoA by another 4. Maybe the numbers don't match because there are some amino acids which can form more than these products, maybe more than one product. Then if they form all this, what do you call them? They are classified as glucogenic. If they form any of these products, you call them glucogenic. What do? They provide energy when it is required and they provide glucose when required. So this is the metabolism of the carbon skeleton. The other 2 products that can be formed. They can also form acetyl-CoA or acetoacetyl-CoA. They are classified as ketogenic and they provide lipids. They can result in the formation of lipids or energy as required. So let us start with the first 2 amino acids which form oxaloacetate. What is oxaloacetate? It is an intermediate of the TCA cycle. So briefly, which are the 2 amino acids which form oxaloacetate? Very easy, should be known because we are talking of aspartate transaminase from so many classes, including its isoenzyme importance. So oxaloacetate, and it comes from aspartate. The most important amino acid which forms oxaloacetate is aspartate. From which is this enzyme? AST, aspartate transaminase. So if aspartate can form oxaloacetate, which other amino acid can form oxaloacetate? Remember, just as the way glutamine and glutamate are there is one more amino acid, aspartate and asparagine. Remember, asparagine, you again ASN. What is it? 1 amino extra group. So asparagine is converted to aspartate. How? Asparginase. Asparagine is converted to aspartate. Aspartate is transaminated to form oxaloacetate. So we have got one intermediate formed from 2 amino acids, that is oxaloacetate. So asparagine and aspartate. So asparginase hydrolyzes asparagine to form aspartate and ammonia is liberated. Aspartate undergoes transamination to form oxaloacetate. Aspartate transaminase. Remember that both these asparagine and aspartate are non-essential. Why? The reverse reaction is also possible. Transamination is always reversible, requires pyridoxal phosphate, and the reverse reaction is also possible. Now let us go to the next important thing that is formed. Now I have given it in a different way. We know that oxaloacetate combines with acetyl-CoA to form citrate. Citrate is converted to isocitrate and then further to form alpha-ketoglutarate. I am talking of certain intermediates of the TCA cycle which are important as far as the entry points of different amino acid metabolism. So please also remember that acetyl-CoA has come from pyruvate. Pyruvate is converted to acetyl-CoA And also remember that pyruvate is also a precursor of oxaloacetate by pyruvate carboxylase, and this is pyruvate dehydrogenase. This pyruvate to oxaloacetate occurred in gluconeogenesis. So the compounds which form pyruvate are also gluconeogenic. Compounds which form the intermediates of the TCA cycle are also glucogenic, whereas the ones which form acetyl-CoA are not glucogenic but ketogenic. So with this background, again we'll go to alpha-ketoglutarate. Which are the amino acids? One amino acid should be known to you, and that is glutamate. Glutamate should be known to you. What else is should be known to you? Glutamine. Naturally, if you have aspartate and aspargine, you have glutamate and glutamine, which can form alpha-ketoglutarate. These go hand in hand. Glutaminase to glutamate, glutamate to alpha-ketoglutarate by the enzyme, any of the transaminases. All the transaminases give to glutamate to form alpha-ketoglutarate. The vice versa, it keeps on happening. Okay, so we have glutamine and glutamate. The other amino acids, I have just listed it down here, include proline. Proline can form, arginine can form, and histidine can form alpha-ketoglutarate. We have to remember them. So you have got proline, arginine and histidine, happiness. So glutamate, glutamine, and these are the other amino acids. Just briefly touching about it, I have told to you how glutamate and glutamine are converted to alpha-ketoglutarate. Please look at these reactions. Glutamine by glutaminase forms glutamate. Glutamate to alpha-ketoglutarate can be by transamination or by glutamate dehydrogenase. Where is this enzyme involved? It is involved in the production of ammonia in the liver. So glutamate dehydrogenase can also result in the formation of alpha-ketoglutarate. Now let us talk about proline to alpha-ketoglutarate. I am just not going to talk to you about individual amino acids. Let us talk of their importance more than what is the pathway. So proline is oxidized to glutamate. How? Glutamate is then converted to alpha-ketoglutarate. So proline forms proline 5-carboxylate, which forms glutamate gamma semialdehyde, which forms glutamate. Again, I have put it in the PowerPoint. I am not emphasizing it because nobody may ask you regarding this. What is important is you should know that proline can form alpha-ketoglutarate. Arginine can form alpha-ketoglutarate. Histidine can form alpha-ketoglutarate. That is important. Arginine to alpha-ketoglutarate, how does it occur? As we all know, arginine was an intermediate of the urea cycle. So arginine can be converted to ornithine by the enzyme arginase, where urea is formed. So arginine to ornithine can undergo a change. Ornithine can bypass the urea cycle, it can go to something else. Ornithine can be converted to glutamate semialdehyde, which forms glutamate and alpha-ketoglutarate. Again, I am not emphasizing the different pathways. I am just telling you briefly so that you will have an idea. That's all is enough. Now the next amino acid is histidine. Histidine, how is it converted? To histidase. This I have already told to you. Histidine to urocanic acid is by the liberation of ammonia. Histidine to urocanic acid is by the liberation of ammonia, and this has come by the action of the enzyme histidase. This is important because ammonia is liberated here. Urocanic acid by multiple steps is converted to form aminoglutamate. Should you know about this by this metabolite? Yes, it is also abbreviated as FIGLU. Form aminoglutamate, FIGLU. What is the importance of FIGLU? Now I'll just tell you, form aminoglutamate can form glutamate. Please remember, when we have got form aminoglutamate to FIGLU to glutamate, we require folic acid. So tetrahydrofolate is converted to N5-formyl tetrahydrofolate. What is this importance of this? The enzyme is form aminotransferase. So what is the importance of this reaction, histidine to alpha-ketoglutarate? Importance is, if folic acid is deficient, what will happen? If folic acid is deficient, this reaction, FIGLU to glutamate, will not take place. So what? Fig-Glu levels will increase in the blood. Is it normally detectable? Maybe to some extent we actually cannot make out. So there is what is called as the histidine load test. So we give a lot of histidine and then you estimate Fig-Glu. What will happen? Fig-Glu, if there is folic acid deficiency, Fig-Glu will not be converted into glutamate. This is mainly of historical significance and MCQ point of view, it is definitely important. So when folic acid is deficient, Fig-Glu is excreted and this is known as the histidine load test. Histidine is given in a big load and you estimate Fig-Glu to know about folic acid deficiency. So that is why you should know about histidine and its conversion to glutamate. Fig-Glu. So which are the amino acids which form alpha-ketoglutarate? Glutamate, glutamine. That is easy to remember. Histidine should be remembered because of Fig-Glu. Glutamate comes in that. So glue. So Fig-Glu to histidine. So you can remember histidine. The remaining two are proline and arginine. Proline and arginine. Arginine has come from urea cycle. So maybe that's how you remember that ornithine is converted to glutamate. So you know that. And proline also is nearly the same pathways actually, arginine and proline. So now we go on to the other amino acids which form pyruvate. Now there are 6 amino acids which form pyruvate. These were 5, these were only 2 and that's why I have gone in increasing number. Which are the amino acids which form pyruvate? All these are also glucogenic. Now one important amino acid that should come to your mind is alanine transaminase. So alanine naturally will form pyruvate. Which are the other ones? Whenever we talk of alanine, there is also glycine and serine. We call that the gas compounds. Glycine, alanine, serine, they are somewhat together. All of these can form pyruvate. Cysteine can also form pyruvate. I'll just give you the list so that you can remember it for the MCQs. So, and one way to remember is glycine, alanine, serine. All three are the gas. Many times they go together. Cysteine and its compound cysteine. Threonine also forms pyruvate. Tryptophan also forms pyruvate. We have got gas and cysteine, threonine and tryptophan. Now briefly, alanine to pyruvate, you already know transamination. So let us talk about serine and glycine to pyruvate. Glycine and serine metabolism go hand in hand. Now I have shown it over here just so that you know that there is involvement of tetrahydrofolate, folic acid in these reaction. Now glycine is converted to serine or serine can be converted to glycine. So this is a 1 carbon metabolism that is taking place here. So glycine to serine, serine by serine dehydratase. Remember the reaction I told you where ammonia is produced, amino acid dehydratase. So serine is converted to pyruvate. That is how serine and glycine form pyruvate. Now cysteine to pyruvate, how does it happen? Cysteine is reduced to gas CTT from pyruvate. Now what is cysteine? Cysteine comes from cysteine or cysteine. Cysteine is reduced to cysteine and then later on metabolized. You need not know the pathway. It is transaminated to form beta mercap to pyruvate and pyruvate. Now importance of cysteine is that can release sulfur as elemental sulfur or a sulfite, and this sulfur is used to synthesize an important compound called as 3'-phosphoadenosine 5'-phosphosulfate. Big name, better known as PAPS. PAPS is involved in the formation of sulfolipids, sulfoproteins, or wherever sulfur metabolism is involved, PAPS is involved. Now we go on to the next one. Cysteine is over. Threonine. Threonine, how is it from pyruvate? It is actually, threonine is just broken down and pyruvate is released. Pyruvate is released. So threonine also forms one more intermediate of the TCA cycle, and that is succinyl-CoA. Okay, I'll just write here succinyl-CoA. Succinyl-CoA. Remember alpha-ketoglutarate. There are multiple steps involved here. I'm just telling you the important ones. Succinyl-CoA. So threonine can form pyruvate and threonine can also form succinyl-CoA. This comes from threonine can actually form propionyl-CoA. Propionyl-CoA to succinyl-CoA. Threonine, pyruvate. Now what is important here is that threonine does not undergo transamination. Remember, lysine, threonine, proline and hydroxyproline do not undergo transamination. So deamination reaction threonine dehydratase forms pyruvate and propionyl-CoA, and it is purely glucogenic. We go on to the next T, and that is tryptophan. Remember, tyrosine is entirely involved with phenylalanine metabolism. There are 3 T amino acids, that is threonine, tryptophan and tyrosine. Tyrosine always goes with phenylalanine, so it is easy to remember what it forms. Threonine and tryptophan go hand in hand. Now tryptophan, again, one important thing that is happening is tryptophan is converted to kynurenine. Different name, you should be knowing the names, that's all. This is called as the kynurenine pathway. Kynurenine further forms 3-hydroxykynurenine to 3-hydroxyantranilate. Now importance is kynurenine can be converted to xanthurenic acid, and for the further metabolism pyridoxal phosphate is required, and plus 3-hydroxyantranilic acid or antranilate is involved in the formation of NAD, and this is further converted to acetoacetyl-CoA. So the kynurenine pathway is both glucogenic and ketogenic. So tryptophan is both glucogenic and ketogenic. So you had threonine which formed pyruvate and succinyl-CoA, whereas you have tryptophan which is forming both acetyl-CoA or acetoacetyl-CoA and pyruvate. So threonine can form these two compounds. Acetyl-CoA, it becomes ketogenic, and it can form pyruvate, so it becomes glucogenic. So 3-hydroxykynurenine to 3-hydroxyantranilic acid, alanine is released, and that is why alanine is converted to pyruvate. This is B6 dependent, and this, remember that 3-hydroxyantranilic acid can enter the NAD. So what is the importance of this is tryptophan can help in the formation of a vitamin nicotinamide adenine dinucleotide. The coenzyme form of a vitamin can be formed. When do we say anything is a vitamin? When it cannot be synthesized, that is what we used to say, but there are certain vitamins which can be synthesized, and one of them is nicotinamide adenine dinucleotide, niacin. So niacin can be synthesized from tryptophan by the NAD pathway, or it can be ketogenic also. So with that finishes pyruvate. Now we'll go on to fumarate. Another intermediate of the TCA cycle. Few means Fe. So, which are the amino acid which form fumarate? Will be phenylalanine. Phenyl, easy to remember. Whenever phenylalanine is involved, you have got tyrosine also along with it. Without tyrosine, we do not have phenylalanine. They go hand in hand. So, both phenylalanine and tyrosine form fumarate. Fumarate is further converted to oxaloacetate, and it goes into the malate to oxaloacetate, which goes into the gluconeogenic pathway. So let us talk about fumarate. Only 2 amino acid, phenylalanine and tyrosine. Now an important reaction, very important reaction, phenylalanine in the presence of oxygen and tetrahydrobioctene is getting converted to dihydrobioctene, forms tyrosine. The enzyme is phenylalanine hydroxylase. The enzyme which synthesizes tetrahydrobioctene from dihydrobioctene uses NADPH, whereas the one which regenerates this uses NADH. So that is the importance of, here I have written in the NADP and NADH, phenylalanine is converted to tyrosine. Important point is phenylalanine hydroxylase requires tetrahydrobioctene, and in the presence of oxygen it converts phenylalanine to tyrosine. Why this is such an important reaction? No phenylalanine hydroxylase, it results in a disease called as phenylketonuria. So that is why it is important. Now what happens further to tyrosine metabolism? There are different steps involved here. I have just written it down. You need not know the pathway. Just remember that there is one intermediate that is formed, that is homogentisic acid. Then finally it forms fumarate and acetoacetate. So it forms fumarate and acetyl-CoA, or a ketogenic portion of it, acetoacetate. This is the metabolism of tyrosine. We go on. Succinyl-CoA I have already told to you. One is threonine. Which are the other ones which form succinyl-CoA? It involves methionine, valine, isoleucine, Vim, valine, isoleucine and methionine, out of which I have written methionine first and threonine, which is anyway formed succinyl-CoA. So let us talk about methionine. How does it form succinyl-CoA? Methionine is converted to its activated compound that is known as S-adenosyl methionine. S-adenosyl methionine is also abbreviated as SAM, and it is further converted to S-adenosyl homocysteine, which is later forms homocysteine. What is required for this conversion? ATP is required for this conversion, and the enzyme is methionine adenosyl transferase. Now, what happens to SAM, S-adenosyl methionine? S-adenosyl methionine transfers its methyl group to an acceptor, whichever wants that methyl group. You give it to that acceptor. That acceptor gets converted to its methylated product. So, S-adenosyl methionine gives its methyl group to the acceptor. Normally, remember normally the sulfur in methionine is in a thioether linkage in methionine. So this sulfur cannot be released normally, but if it is activated, that is, it forms S-adenosyl methionine. When an adenosyl group comes and attaches to this sulfur, this methyl group gets, okay fine, I can go away from here, and it can go to a acceptor and it is converted to its methylated product. So that is the importance of S-adenosyl methionine. Methionine can form succinyl-CoA. So we have valine, isoleucine, methionine which can form succinyl-CoA. Now when it gives out its methyl group, it forms S-adenosyl homocysteine. If you remove that adenosyl group from that, it becomes homocysteine. So S-adenosyl homocysteine to homocysteine. Now further fate of homocysteine, I'll talk about it in my next session. What happens to this homocysteine that is formed, and let me also talk about the other remaining ketogenic and other amino acids in the next session. Thank you.